system is fallopian tube. Inside the fallopian tube there is one region that is called ampulla. In ampulla region we can see the fertilization. If you see the female reproductive system, this is the female reproductive system. Female reproductive system. These are the ovary. Okay, this one is a uterus. The ovum is released from the, this is the ovum which is released from the ovary. After that this ovum is enter into the fallopian tube. This is the ampulla region of fallopian tube. In the ampulla region of fallopian tube you can see the fertilization. Because of insemination what happens? The male gametes or sperms are enter into the female genital tract. Finally that sperms are reached to the ampulla region of fallopian tube. There we can see the fertilization or fusion of both male gamete and female gamete. Next, here when the fertilization occurs, if both male gametes and female gametes move to the ampulla region simultaneously, then only you can see the fertilization. If male gamete move into the ampulla region at one time and the female gamete move the ampulla region at a different time, in that condition we cannot see the fertilization. Okay, how the fertilization occurs we have to see now. See, fertilization. If you see the ovum, this is the ovum which is in secondary oocyte stage. Secondary oocyte. Okay, this is the secondary oocyte or ovum which is in secondary oocyte stage. Okay, it is mainly covered by two layers. It is mainly covered by two layers. What are that main two layers which are present surrounding to the ovum? That are, number one is corona radiata. Next, number two is zona pellucida. Corona radiata is outermost layer of the secondary oocyte or ovum which is in secondary oocyte stage. Then zona pellucida is innermost layer. Okay, when sperm reach, this is the sperm. When sperm reach to the ovum, the acrosome, this is the acrosome of sperm. The acrosome of sperm will release two enzymes, mainly two enzymes that are hyaluronidase, next acrosin. The hyaluronidase enzyme can cleave the corona radiata. Next, the acrosin enzyme can cleave the zona pellucida. After that, what happens? The sperm is enter into the secondary oocyte. Okay, this is called fertilization. Here already we know that in each ejaculation the males can produce approximately 200 million sperms. Here only one ovum is produced during the each menstrual cycle, at middle of each menstrual cycle. But only one sperm can fertilize the one egg. Here what happens? When first sperm, during the fertilization, when first sperm enter into the ovum, there is a contact in between the sperm and the zona pellucida of ovum. During fertilization, when sperm contact with zona pellucida of ovum, the zona pellucida layer involving physiological changes. Okay, after the physiological changes, the zona pellucida will not allow the further entry of another sperms. Hence, because of this, only one sperm can fertilize the egg. Okay, this is about the fertilization. After fertilization, we have to see about the formation of zygote, then how the zygote is becoming to embryo. Then we have to see about the embryonic development. Okay. Welcome to Lakshman Biology. Now we have to discuss about the embryonic development. Okay, in embryonic development, welcome to Lakshman Biology. Now we have to discuss about the embryonic development. After fertilization, what happens? We can see the formation of zygote inside the fallopian tube. See, This is the female reproductive system. This is the fallopian tube. This is the fallopian tube. This is the uterus. These are the ovary. Okay. Next, if you see, fertilization occurs at ampulla region. The fertilization leads to formation of zygote. This is the zygote. Zygote. Again, after the formation of zygote, this zygote will move from the ampulla region of fallopian tube to the uterus. While moving the zygote into the fallopian tube, the zygote is involved in different changes by the cleavages. Cleavages are one type of rapid mitotic divisions which leads to formation of more cells inside the zygote. Okay. While formation of embryo or while movement of the zygote from the fallopian tube, from the fallopian tube to the uterus, which is involved in cleavages. Already I told. Okay, because of cleavages, what happened? The one cell zygote become into two cell stage. Again, the two cells become into four cells become into 8 cells. Okay, the newly formed cells which are present in the embryo are called blastomeres. Okay, the embryonic stage which contain 8 to 16 blastomeres is called morula. Here what happened? Because of cleavages, first zygote become into an morula. Morula cells again rapidly divide and then become into an blastula or blastocyst. Okay, blastocyst is formed from the morula. Listen once again, zygote involved in cleavages and become into morula. Morula again become into blastocyst. Okay, if you see the structure of blastocyst, this is the blastocyst. The structure of blastocyst mainly contain two layers. The cells which are present in the blastocyst arranged in two layers. Outermost layer of blastocyst is called trophoblast. Next, innermost cells, inner cell mass. Inner side we can see the cells inside the blastocyst. This layer is called inner cell mass. Okay, trophoblast is outermost layer, inner cell mass is a inner layer. This is about the blastocyst. After that, what happened? This blastocyst is attached on the wall of uterus. The trophoblast layer of the blastocyst attached on the wall of uterus. This is the wall of uterus. The trophoblast layer will attach on this wall of uterus. This attachment is called implantation. What is the implantation? Attachment of blastocyst on the wall of uterus is called implantation. After that, the trophoblast layer will form finger-like projections into the uterine wall. These finger-like projections are called chorionic villi. These chorionic villi are finger-like projections which are used for the attachment. After that, uterine tissue will grow and the blastocyst is embedded inside the uterine endometrium. Okay. Next, after that, what happened? The chorionic villi of trophoblast or embryo combine with the uterine tissue and form the structural and functional unit in between the developing embryo and the maternal tissue, that is called placenta. Now we have to discuss complete details of placenta. What is the placenta? After the formation of chorionic villi, these chorionic villi combine with the tissue of uterus and become into an placenta. Here we have to discuss about the placenta. Placenta is mainly used for the exchange of substances in between the developing embryo and mother tissue or maternal tissue. Mainly it is used for the removal of carbon dioxide and excretory products from the embryo. Apart from that, this is also used for the transportation of oxygen into the embryo, to the developing embryo. That is about the placenta. Okay, next, placenta also used for the production of hormones. The hormones which are produced from the placenta are HCG, HPL, estrogen, progesterone. These are the different types of hormones which are produced from the placenta. HCG, HPL, estrogen, progesterone. HCG means human chorianogonadotropic hormone. HPL means human placental lactogen. Then estrogen and progesterone. These are the 4 different hormones which are produced from the placenta. Okay. After that, what happened? The blastocyst again developed into an embryo, and that finally at after 9 months these blastocysts completely developed into an baby. Okay, the 9 months pregnancy of females is divided into 3 trimesters. First trimester contain 3 months, second trimester contain remaining 3 months, next 3 months, and third trimester contain last 3 months. Okay, 9 months duration of pregnancy divided into 3 trimesters. First trimester 3 months, second trimester 3 months, and third trimester 3 months. Okay, after that what happened during the first month? How the embryo is developed? At first month you can see the formation of heart. Heart is formed at first month.